Let's tackle the middle section, section B, sometimes the most intense for students, especially if they have some issues understanding molecular biology and chemistry. The scope of the textbook, and thus this video, is outside the full remit of what the kidney can handle. It's a fantastic organ, so we get a chance to look at some of the more salient features. Water is the most important commodity for land-based creatures, and the management of that water is of the utmost importance. We have to balance what we take in with what we output. Figure 14.3 gives us an average for an average human adult male. So the intake, roughly on a daily basis, should be about 1.4 liters in liquid form, 1.1 liters in food in various forms, and about 350 milliliters is produced by condensation reactions involving molecular rearrangements. The total thus comes to just under 3 liters a day. The output has to be balanced. So people assume that most of the water is lost through the urine, and they would be correct. So of the approximately 3 liters, about 2 liters is lost through urine. But there is also other losses in the body. The second most obvious is sweat, but that only accounts for about 50 milliliters a day. We also lose a tiny fraction in the feces, about 100 milliliters a day. And believe it or not, the lungs and the skin combined will lose almost a liter in themselves because we have to moisten our breath in order to make the lungs more efficient. The second most important commodity is sodium chloride, sodium in particular. Salt intake is about 8.5 grams for an adult male, and the output therefore has to balance. Most of the salt is lost through the urine, but a small amount is lost through the feces as well as through sweating. If the salt content of a human body changes, there's a short-term adjustment in the pool of salt internally, and a new homeostatic point is set, a new set point. But eventually, if the diet is modified back to the original, that additional salt will over a number of days be reduced with the equivalent loss of water. As we proceed over the next few slides, looking at how the different ions are balanced, it's important to relate back to this figure here. This is a two-dimensional view of a kidney nephron and its blood supply. So even though not realistic, it's enough to allow us to understand some principles. The first thing is that blood enters and interacts with the Bowman's capsule first via the glomerulus. Then the blood moves on and travels along the loop of Henle. It does a U-turn at exactly the same location as the loop itself, and then the blood flows back up before exiting this region of the kidney. At the same time, the nephron, this part here is the proximal convoluted tubule. This is the descending part of the loop of Henle. This is the loop of Henle itself. More importantly, the ascending loop of Henle has two major regions. The constricted portion here, the thin part, and the thicker part above. Then we have the distal convoluted tubule, which is then followed by the collecting ducts. And the collecting ducts make a journey again down the entire length of the loop of Henle before continuing into the pelvis of the kidney. The biggest takeaway in terms of anatomy is that this form has been evolved over many eons of time to do exactly what it needs to do. It needs to be able to make urine between two different ranges. If the animal is really hydrated, then it needs to eliminate most of that water without losing a lot of the ions and other minerals. If the animal is dehydrated, then it needs to preserve as much water as possible, but still continue to get rid of the toxic materials that it has to. So here we go. Let's begin with sodium. one of the most important electrolytes in the body. The tubule has the ability along its length to allow its cells to absorb sodium from the filtrate. This occurs through active processes. The only region unable to do so is the descending loop of Henle. So this region here, the cells lining this part of the tubule are prevented and therefore unable to move sodium in either direction. And that's a very, very important fact for you to understand. The next important observation is that water reabsorption from the tubule is by osmosis. And as we know, osmosis is a passive process involving diffusion of water across a membrane. But the driving force is dependent on the reabsorption of sodium. So if sodium moves, water will tend to follow. For most cells, water can move through the membrane itself, believe it or not. Even though it's a polar molecule, it's so small, it has the ability to filter its way by bombardment through the phospholipid bilayer. So enough water can pass through a plasma membrane. But in other cases, when large quantities of water are required to be transported from one side of a membrane to the other, you need to have special pores, channel proteins. And one of the most successful is the aquaporin channels. These are giant protein donuts through which water can pass if the donut is open. So the important thing is that these aquaporins can be regulated. The simplest way to regulate these aquaporins is to either remove them from the plasma membrane by invagination, endocytosis, or to introduce them into the plasma membrane in quantities by exocytosis. The cells which can do this are directed to do so by external signals. And the most common external signal is by a hormone called antidiuretic hormone, or vasopressin. Unfortunately, it has two names, the old name ADH and the more current name vasopressin. Sorry to confuse you. It is the influence of this hormone that directs the presence of aquaporins at greater numbers in the membranes of certain cells. The cells which are most likely to have that capability are located here in the collecting ducts. Let's turn our attention again back to sodium and see how sodium is regulated in the proximal tubule. The proximal tubule, once again, will be in this region here. It's also called the proximal convoluted tubule. Figure 14.14a gives us a schematic of these proximal tubule cells which are lining the tubules in that part of the nephron. So this is the apical surface and this is the basolateral surface of these cells. There's the tight junction and this will be the interstitial fluid, and then the blood vessels will be over here somewhere. This particular surface has in it co-transporters and countertransporters, symports and antiports. Here we have the sodium-potassium pump, and in this case we also have potassium ion channels and some other types of proteins that can regulate the concentration of substance X. The primary purpose here is to show how sodium is pulled out of this fluid into the interstitial fluid. On this side we have a pump. That pump keeps the sodium concentration low inside the cell and high inside the interstitial fluid, and that uses a lot of energy from food molecules. Because the sodium concentration is low inside the cell, all you have to do is allow these transporters to be activated and they shall effortlessly move sodium from the lumen, from the filtrate, into the cell and from there into the interstitial fluid. Therefore, sodium is removed from the proximal convoluted tubule by the process of reabsorption, and that's explained at the molecular level. Slide 14.14b again addresses sodium reabsorption, but in a different location, in different class of cells. Where are those cells located? Let's go back to the figure, and those cells will be located in this region here. How do they do it? They do it using a slightly different mechanism. What's common is that the sodium-potassium pump continues to work, and that keeps a low level of sodium inside the cell. The difference now is that the alternative types of transporters are activated. So for the first time, we have a sodium chloride transporter and a potassium transporter. If you look over this schematic, you'll notice that the recycling of potassium drives the recovery, the reabsorption of sodium into these cells. Something different from 1414A. Let's compare these two locations to a third. Figure 1414C looks at sodium reabsorption, now in the cortical collecting ducts. So where are we now? We're here, in this region. What's happening there? We can see that the mechanism here is again slightly different. What's common is the sodium-potassium pump, which keeps a low level of sodium inside the cell. But in this case, we just have two simple ion channels. The outward movement of potassium down its gradient drives the inward movement of sodium down its gradient. So the exchange of positive charge on either side is important because that charge is balanced. There's no net change. And we'll talk about that in more detail in the chapter on nerve impulses. The electrical force may be balanced, but the chemical differences are great enough that each ion moves according to its own concentration gradient. And this, once again, achieves the reabsorption of sodium from the filtrate into the interstitial fluid. Please take your time to understand and maybe explain to somebody else this comprehension. Figure 1415 looks at the combined effects of sodium and water reabsorption. As we said earlier, once you move the sodium and you allow the membrane to move water, then the two will travel in the same direction, with the sodium pulling the water across. This is indicated in this figure quite nicely. So once again, there's a pump. That's your sodium-potassium pump, and it's keeping the concentration of sodium inside the cell low. The sodium will move down from the apical surface if those ion channels are open. And as the sodium moves, that will cause a change in the local osmolarity. So the water concentration here will tend to increase, and the water will then travel by osmosis into this cell. And in some cases, the water molecules are so tiny they can breach the tight junctions and end up, in both cases, entering the interstitial fluid. So water is moving because of the activity of sodium. Figure 1416 supplements our understanding of the aquaporine discussion that we had a few minutes back. These cells are those special cells that are located in the collecting ducts that have the ability to regulate the concentration of aquaporins that they have. So this vesicle contains a lot of aquaporins, but they are not in use. They're deep inside the cell. These aquaporins are on the plasma membrane, and they will be in use. So the more of these you have in this location, the greater the amount of water that can be moved due to concentration. If you look carefully in this example here, there's a very sophisticated movement of water from the lumen of the tubule through the cell and into the interstitial fluid. The mechanism influencing how many aquaporins will be present in the apical membrane is the hormone ADH, or vasopressin, and that's indicated here in purple. So if vasopressin is present in this vicinity, it will bind to its receptor, activating the receptor and causing signal transduction within the cell. The vasopressin receptor activates an enzyme, adenylate cyclase, and that enzyme takes ATP and causes it to be reconfigured into cyclic AMP. Cyclic AMP is a very powerful internal messenger inside the cell, and that drives the phosphorylation of membrane fusion proteins, very similar to the snares, which direct the vesicles to fuse with the plasma membrane, thereby automatically increasing the number of aquaporins available to transport water across the apical surface. We will revisit some of these alternative mechanisms of transporting solute later on. Let's turn our attention to the overall effect that the structure, the anatomy of the kidney nephron and the blood vessels, the Vasa recta, impart on that tissue in that part of the kidney. The mechanisms outlined on this slide explains how this gradient is generated. That's very important, regulating the output of different types of urine. Students should watch the video on countercurrent multipliers, especially with respect to the kidney. That will help understand this section. There are 5 main driving forces creating the hyperosmotic medullary interstitium. Interstitium means the tissue outside the tubule and outside blood vessels. Medullary is the location in the kidney, and hyperosmotic means very, very salty. The first of these is the arrangement of the structure of the tubules, and we showed you earlier that this arrangement and this arrangement, these loops at the bottom, are very important in maintaining this background color. You can see that the slide goes from a lighter brown through a medium brown to a darker brown. And that's what we're trying to explain, how that gradient is established and maintained in every single kidney. The countercurrent multiplier will help with that understanding. The second component is the reabsorption of sodium in the ascending limbs of the loop of Henle. And those ascending limbs have been evolved to such a point that they are impermeable to water. So only sodium can leave the tubule, and if water wants to follow, it cannot. Consequence is that in that region, and we're looking at this region here, as the sodium leaves, the filtrate becomes less and less concentrated and solute, i.e. it becomes more and more watery. And that process continues until you get to the top of the ascending loop of Henle, and here the filtrate is even more dilute than the plasma or any other fluid in this region. The fourth factor is the concentration of urea and how it's trapped in the tissue. So trapping of urea in the medulla is going to be also responsible for maintaining this hyperosmotic region. And then finally, the hairpin loops of the blood vessels, known as the Vasa recta, they also have to have this hairpin structure overall in order not to wash away the gradient. That's important. Had the blood vessels been arranged in a linear fashion going up and down this page, any gradient generated by the tubule would immediately be washed away by the flow of blood. So the blood vessels, which should be indicated here alongside, would follow the tubule in its design. So the bottom part would be a loop, and that would be the loop of the Vasa recta. Again, that's the reason why this blood vessel here is making a U-turn, just like the loop of Henle. Once again, the video helps understand the next 2 slides, but if you have no access to the video, try to follow through. So we have here the loop of Henle. This is the descending limb, and this is the ascending limb. The important thing to understand is that only sodium is able to be transported from the filtrate into the interstitium. Water cannot penetrate across these walls, at least here. On this side, only water can move and not sodium. So any sodium inside the filtrate will remain. The consequence of this is that as the filtrate travels through the hairpin, it loses water from this limb at a high rate, and it becomes more and more concentrated in solute. So these numbers refer to the osmolarity. The value for plasma and blood is 300 millimolar. Because of the activity of these cells on both sides, the movement of water here and the movement of sodium there causes this hairpin to eventually generate this gradient. And you can see down the bottom, the number's not 300, it's actually close to 1400. This fluid here, the filtrate, is 4 times or more concentrated than that of blood. As it makes the U-turn, the same fluid, as it begins to travel up the ascending limb, undergoes a reversal. The pumping out of massive amounts of sodium results in a decrease in the osmolarity, and eventually near the tip, so much salt is removed by active transport that the remaining water becomes more and more concentrated, and that becomes even more dilute than blood. The net result of this molecular activity of different types of cells produces that gradient from the top down to the bottom of the hairpin. This gradient is used to great effect to condition the production of urine as it then flows through the collecting ducts. In this example, the lowest number that we have is 80 milliosmolar. If that is not then altered in any way by the cells lining the collecting duct, then the same fluid will be eventually secreted from the bottom of the collecting duct into the renal pelvis. That would be very, very dilute urine. So that's how you make dilute urine. You just shut down this particular avenue. However, if you have some water concerns, then what you can do is allow these cells to continue to be influenced by the medullary gradient. They therefore open up and they then allow much of this water to enter the tissue. Then as the filtrate travels down here, it becomes almost as concentrated as the interstitium. And down here it'll be 1400 milliosmolar. Very, very thick urine. And therefore you can then, in those circumstances, produce a thick, concentrated urine. So between this range and this range, you have the full spectrum of the type of urine that land vertebrates can produce. One compound takeaway message from this section is that if we drew a line down here that intersected at this point, everything on this side of the line with respect to the nephron would be automatic based on the speed of flow of the filtrate. Nothing else. Everything on the other side of that line, i.e. this part of the distal convoluted tubule and the collecting duct, would be under regulatory control. Their adjustments can be made. So when filtrate is first produced, it undergoes general conditioning until it gets to this point, and after that, then the conditions within the body, other than blood pressure, will decide how and by how much that filtrate is adjusted. Combining our knowledge so far with respect to the kidney, we can say that the first part of the nephron just conditions and makes very hyperosmotic filtrate, and the second part of the nephron is responsible for making some adjustments, and one of those chemicals, the hormone ADH or vasopressin, can make the collecting ducts permeable to water to different degrees, therefore influencing how much water is sucked out of the filtrate. This slide reinforces that exact same concept. Here we have ADH. On the previous slide we had low levels of ADH or absent ADH. You should now be able to predict the type of urine in terms of its concentration that's produced across the range depending on the activity, at least for this substance here. Sodium is not the only commodity regulated along with water. Many other substances like potassium and hydrogen and chloride, they are all adjusted along the length of the nephron. One other chemical, urea, the waste product of metabolism, is pretty toxic, and thus its elimination is favored. But when we do the analysis on the way that urea is handled by the nephron, it's interesting. Of all the urea that is filtered through the glomerulus and Bowman's capsule, 50% is reabsorbed in the proximal convoluted tubule and is reabsorbed into the interstitium. The remainder then passes down the loop of Henle and then back up into the collecting ducts, and another 30% is reabsorbed in the collecting ducts. As it passes through the lower reaches of the collecting system, more is reabsorbed. So we can see that the vast majority of the urea is reabsorbed away from the filtrate. About 15% is then eliminated in whatever concentration of urine is produced. This calculates to 85% of the filtered urea is then reabsorbed. Most of that stays behind in the interstitium, and it maintains this high salt concentration, solute concentration, interstitial gradient. Some of it is then removed back into the blood, about 5%, but the vast majority contributes to the gradient. Please take a few minutes to comprehend the meaning of these two graphs. They're talking about ADH, vasopressin, and how it can then regulate the concentration of the filtrate by its activity. In the upper graph, we see the locations along the tubule. So this is the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, the cortical collecting ducts, and the medullary collecting ducts. As the filtrate travels through this system, its volume, if we assume that the filtrate at the glomerulus is 100%, its volume begins to reduce rapidly, and when it comes through the loop of Henle, its volume stabilizes, and it continues to then decline, but that decline is impacted by either the absence or the presence of vasopressin. So if no vasopressin is present, then there is no further exaggerated removal of water from the filtrate, and eventually the filtrate that's produced will be about 12%. If vasopressin is present at maximum levels, the cells in this region, they continue then to have activity, and that's the difference between the red line and the blue line, and those cells will remove a lot of the water because of the presence of vasopressin, and the filtrate will therefore be minimized in terms of volume. This correlates back to what we said earlier, that in the absence of vasopressin, the fluid is diluter, and in the presence of vasopressin, the filtrate, the fluid in the filtrate is more concentrated. The bottom graph likewise has the same x-axis. These are the same regions along the tubule, but this time we're looking at the osmolarity, the strength of the fluid. So blood is about 300 milliosmolar, so that'll be the same concentration. The filtrate here will be the same as blood, and it doesn't do anything until it gets into the loop of Henle. So let's follow the red line. So in the absence of vasopressin, that fluid becomes slightly more concentrated, and then it becomes hyperosmotic. It becomes more dilute than the blood itself, and then when it's exported out of the collecting ducts into the pelvis of the kidney, it's very, very dilute. On the other hand, in the presence of vasopressin, as the filtrate enters the loop of Henle, it's then influenced by the interstitium, and it becomes it becomes hyperosmotic to 1200 milliosmolar. Then up the ascending limb of, then as it travels up the ascending limb of the loop of Henle, it then begins to become more and more dilute until it reaches the same point as the fluid in the absence of vasopressin. So up to this point, the two would be exactly the same. But after this, the effects of vasopressin on the cells in the cortical collecting ducts are then exposed. And from this point onwards, these cells respond by moving water out of the filtrate and therefore making the filtrate, the urine, very, very concentrated. The next few slides are cause and effect. These are feedback loops. If you go back to chapter 1 on homeostasis, you will understand more fully how this system works. But let's do something very similar for each one of these slides. Let's look at the condition at the top and then see what the result is at the bottom, and then understand which mechanism within the body is responsible for making that change. Slide 1422 states that the patient has diarrhea. So with diarrhea, there's an increase in the loss of sodium and water, and the body has to then compensate, otherwise dehydration and death may ensue. So the end result is to preserve the sodium and the water loss. If you uncover the system, you can see that the kidneys are involved, as well as blood pressure and the vascular system, and sometimes the heart, and there's some detectors in various places inside your arterial system and your brain. All these systems are designed to monitor different commodities within your body in terms of electrolytes and fluid. Let's try to follow through with the logic. Diarrhea causes a reduction in the plasma volume. Plasma volume then has a knock-on effect in the decrease in the venous pressure. So in the veins, blood pressure drops. That reduces the return of blood to the heart, the venous return. That then has a direct effect on cardiac output. If you've covered the heart already, this should make more sense. If not, you may want to bypass this section and come back later. As the venous return dwindles, the atrial pressure drops. That then has two knock-on effects. We'll follow this one down. There's a reduction in the ventricular end diastolic volume. That reduces the stroke volume. That has a knock-on effect on reducing the cardiac output, which on the other side of the heart reduces the arterial blood pressure. That reduction in blood pressure is important because it has a direct effect on the pressure in the capillaries of the glomerulus. What's happening in there is that systemic drop in blood pressure has the same effect here. Therefore, the amount of filtrate that's produced is reduced. Reducing the amount of filtrate means that the kidneys are now processing less fluid. Therefore, there's an opportunity to condition that fluid and keep most of it in the systemic circulation, therefore reducing the amount of sodium and water that's eliminated in the urine. If we follow a couple of other branches that were generated, let's go back to this one. The reduction in blood pressure causes a reduction in venous pressure. That, in addition to having the effect on venous return, it has a direct effect by some reflex mechanisms in increasing the activity of renal sympathetic nerves via the brain. And this pathway here is only applicable if you've covered the chapters on, again, the cardiovascular system. Regardless, the increased activity in the sympathetic nervous system causes the constriction of the afferent arterioles in the renal corpuscle, and that constriction reduces the flow locally of fluid, blood, through the glomerular capillaries, having the same outcome, a reduction in the amount of filtrate produced. There's one more branch that we didn't cover, and that means, so that refers to this here, a reduction in the arterial blood pressure is also detected by receptors that are embedded in various locations around the aortic arc and up into the carotid arteries, and they have the same effect, and the outcome is exactly the same. So these four mechanisms all work together to reduce the filtration rate of the glomerulus, therefore reducing filtration of fluid. To fully understand figure 1423, it's recommended that you've done the respiratory chapter as well as the cardiovascular chapter and the circulation chapter, but maybe not the chapter on digestion, because the influence of the liver is minimal here. What you have to understand is what's going on. This system refers to the interplay between the kidney, the heart, the lungs, the liver, and then back into the adrenal glands before the kidney is influenced. In my opinion, other than the nervous system, this is a wonderful interplay between various organs throughout the body where there's a division of labor. Different organs and their tissues are performing different functions for the common good. This particular scenario, something triggers certain cells in the kidney to release an enzyme called renin. The renin then enters the bloodstream, where it's going to tackle a molecule called angiotensinogen. It's a peptide of 453 amino acids produced by the liver and then sent into the circulation. Angiotensinogen will then be digested by the renin enzyme into angiotensin 1, which is just a 10 amino acid remnant of its former self. That becomes a semi-active component, and as it passes through the lungs, the endothelial cells of the lungs, they produce another enzyme called angiotensin converting enzyme, ACE. And that ACE enzyme converts angiotensin 1 into angiotensin 2. Now angiotensin 2 loses 2 of its amino acids compared to angiotensin 1, and it becomes the active ingredient. It then travels to 2 destinations. The first is the adrenal gland. The adrenal glands are sitting on top of the kidneys, but they're independent of the kidney. The adrenal gland will then release aldosterone, a hormone, and the aldosterone will then go back into the kidney and influence the behavior of the kidney and cause the kidney to retain sodium and water. Angiotensin 2 also has an effect on the cardiovascular system in that it causes vasoconstriction. And that vasoconstriction will result in direct increase in blood pressure. So both these effects will preserve blood pressure. So you can see renin is a system designed to increase blood pressure when blood pressure drops. We can take the information on the previous slide and put it into a feedback loop in a flow diagram. So this is a negative feedback loop because what happens here, this system counteracts and restores the set value as much as possible. So students should be able to relate to this by going back to this previous diagram. Turning our attention to a slightly different scenario, in this case, the trigger is an increase in plasma volume. Maybe the blood pressure goes up. So what we want to do is reduce the blood pressure, and one way of doing that is to increase the excretion of sodium. Remember, where sodium goes, water follows. And as water is eliminated from the body, then the plasma volume will automatically be decreased. So let's see if we understand this. And then once again, if you covered the cardiovascular system, this will make more sense. The increase in plasma volume is detected by a distension, a stretching of the cardiac atria. Those atria have certain cells that can release a hormone. That hormone, ANP, is a protein which is then released into the circulation. So in the blood, the level of ANP increases. And as it travels around the body, it has a direct impact on 2 locations. The first is the kidneys. And within the kidneys, it causes the arterioles, the affluent arterioles, to dilate. and at the same time it causes the efferent arterioles to constrict. If we go back to a previous slide that illustrates this, that may make more sense. So let me find it for us, and here it is. So what that AMP does, it causes one side to dilate and it causes the other side to constrict. So I'll leave it up to you which of these combinations that achieves. The result is an increase in the filtration rate of the glomerulus, and as more filtrate is produced, then it gives the cells of the tubule more chance to secrete sodium from the plasma into that fluid. There's another branch here, and that branch simply tells us that ANP has a direct effect on decreasing the concentration of aldosterone in the plasma, and as we saw on a previous scenario, that leads to a decrease in the reabsorption of sodium from the filtrate. Therefore, the amount of sodium that ends up in the filtrate and then the urine has increased. Both of these will then reduce the plasma volume of the blood. Figure 1426 begins with a scenario where we have extra consumption of water. So the end result should be an increased excretion of water through the renal system. And you can then follow through as to how that is detected and then acted upon to cause this final effect. In this last scenario, we bring into play the brain and the pituitary gland, which is the master endocrine gland in some people's opinion. Let's put the scenario in place. The patient is seeing a reduction in the plasma volume, a decrease in blood pressure. That can be dangerous. It can cause fainting and death. So the body has to preserve water to maintain the blood pressure. So maybe this person is experiencing heat stroke, for instance. So let's go through and follow what happens. As the blood loses plasma, this is detected through a system where the venous return, the atrial return, and the arterial pressure begins to drop. These pressures are detected by baroreceptors. Baro means pressure, and these detectors are positioned in various places, as we mentioned earlier. And they report back through to the posterior pituitary gland, which has the ability to increase the secretion of vasopressin, the hormone. That hormone then travels through the plasma to the collecting ducts in the tubules, and it increases their permeability to water. So more water is pulled out of the filtrate by reabsorption. That reduces water loss and therefore preserving blood pressure. We'll leave the students to interact with this flowchart and see if they can figure out what's going on. Moving to the next figure, this is a compound perspective on various systems that are designed to overlap and detect particular homeostatic conditions, and then to report to sensor cells. The sensor cells then perform an action that counteracts the change. The winning outcome will be the one that has the higher hierarchy in terms of physiological importance through evolutionary pressures. Sodium, water, urea may be important commodities, but potassium is also an important ion within the body whose regulation has to be within certain parameters. This simplified drawing reveals that potassium is secreted to some degree, almost fully reabsorbed, and then secreted by a different part of the renal pathway. And the regulation takes place between these 2 points. One can see that in this particular pathway regulating the concentration of potassium, the hormone aldosterone once again plays a role in controlling. Figure 1432 illustrates the dual effect of aldosterone on the reabsorption of sodium at the same time as increasing the secretion of potassium. This brings us to the end of section B. If students have been paying attention, then they should be in a very strong position to be able to address most scenarios relating to renal, iron regulation, and water balance. Section C of the chapter examines the effects of the renal system on the pH of the bodily fluids and therefore other tissues. Additional hormones traveling in the blood can influence different cells along the nephron. One thing to learn, in addition to what we learned in the previous section, is that calcium reabsorption is triggered by the concentration of parathyroid hormone in the plasma. The parathyroid hormone influences the distal convoluted tubule and the early collecting ducts in the cortical region in reabsorption of calcium. Likewise, phosphate is an important commodity, and its reabsorption is also under the control of the parathyroid hormone, but at a different location in the nephron. This happens at the proximal convoluted tubule. Table 14.5 does a great job of showing us the relationship between the different segments of the nephron and the physiological functions that those regions perform to a major degree and what factors they control. So students should spend some time relating back to this once they've learned the bulk of the knowledge. Based on our acquired physiological understanding of how the kidney works, we've been able to develop drugs called diuretics that help patients control different aspects of their filtrate. Depending on the drug available and how it functions, we can impact different regions of the nephron. pH of the bodily fluids and tissues is one of the most controlled aspects of human physiology. Variation in the pH, either one way or the other, around the set value can be devastating and cause death. Table 14.6 highlights where gains of hydrogen ions may come from and also where losses of hydrogen ions may take place. One of the unsung heroes of the human physiological system are the buffers. These are normally protein-based or some type of chemical which absorbs either acid environments or basic environments to try to preserve the pH and stop it from fluctuating in a great way. Inside cells, the major buffers are proteins as well as the phosphate molecule. Outside the cells, the most important buffer system is the sodium bicarbonate carbon dioxide system. Kidneys, in association with the lungs, are the major organs regulating pH. The way the kidneys do it is very similar to the way that is done in the lungs. It relies on the use of bicarbonate and the movement of protons. So carbonic acid is an important intermediary, and so is the enzyme carbonic anhydrase. Without the presence of carbonic anhydrase, the production of carbonic acid will be too slow to aid this system. The interchange between carbon dioxide, carbonic acid, and bicarbonate and the protons is an interesting way in which the body can regulate pH. The indirect pathway shown here generally leads to the overall reabsorption of bicarbonate. So here is a bicarbonate, and through this complex interplay of chemistry, what really happens is that the amount of bicarbonate here is reduced and is produced through a novel way over here. So in reality, it's being moved from here to here at the gross level. With this second scenario, we're not moving the bicarbonate, we're making bicarbonate and achieving the results that way. So we don't have any bicarbonate here, but we do produce bicarbonate inside these cells, starting with water and carbon dioxide, and we release that into the plasma. A consequence of that is the production of these protons, and they have to be dealt with, otherwise the inside of the cell would become very acidic. And those protons are eliminated, and then they combine with other material in the filtrate. There are other more complicated mechanisms for adjusting the concentration of bicarbonate in the plasma, and in this case we're looking at the metabolism of amino acids and the elimination thereof of ammonia. There are other more complicated mechanisms for adjusting the concentration of bicarbonate in the plasma, and in this case we're looking at the metabolism of amino acids and the elimination thereof of ammonia. One would expect evolutionary adaptations to cater to both these conditions, and that's what you see on this slide here. So somebody who is becoming more and more acidic will have this type of possible response. Somebody who's becoming basic will have this type of response. See if you can understand along this slide. A nice relay of that information is provided in table 14.8, where it talks about metabolic and respiratory changes. Any condition which affects the functioning of the kidney can therefore be devastating to overall health. Some diseases which affect kidneys are listed here. So bacterial infections were quite common, UTIs, hypertension, high blood pressure over a period of time, and diabetes. End-stage renal disease is one of the main causes of death in the world, and of course these days a great industry when it comes to transplants. This concludes our journey through the renal chapter. I hope that you got a sense of how complicated, but at the same time how satisfying and wonderful evolutionary adaptations of cells have reached. Thank you.